Welcome to the Department of Entomology at the National Museum of Natural History. Here in entomology, we're responsible for insects as well as arachnids and myriapods. We have over 35 million specimens in the collection, covering more than 400,000 species. And we have curators from multiple agencies, including the Smithsonian, as well as Department of Agriculture and the Department of Defense, working cooperatively to curate this massive collection. We work on lots of different things. We work on insect pests of agricultural crops. We work on ecology, biology, conservation-related issues, as well as describing new species to science. So like most entomological collections, the bulk of our collection is also stored, dried, and on pins. However, we also have a lot of material housed in jars and vials of alcohol. These are for immature insects as well as some non-insect arthropods that get too brittle if they're not kept moist in fluid storage. We also have significant holdings of microscope slides, and these are insects that are truly microscopic or dissections of larger insects, since that's part of what we do in the research part of the building. And then we also have a significant number of things that are housed in envelopes, and these are our dragonflies and damselflies, Odonata, and they're entirely stored flat and on cards in glassine envelopes. And this is fairly unique to our collection.
Part of our work in the Department of Entomology is doing routine specimen level digitization of our collection. The program we use to record our specimen inventory is called EMU. In this program, we can input the barcode, we can put information about what kind of insect it is, how it was prepared, where it was collected, and any other information that somebody from the public might need to use regarding the specimen. Each specimen in our collection gets a unique barcode number. On pin specimens, such as these, the barcode is on the underside of the pin, underneath the other labels. For alcohol specimens, they're at the bottom of the vial, so someone can turn the jar over and see all of the identifiers. For slides, we cut the label small and glue it onto the slides so that somebody can find that information. And for our envelope collection, which is our dragonflies and damselflies, the barcodes are on the back of the card. And these records we put into EMU, where we can put all the information from the labels, the information about the specimen, and how it's prepared. And by doing this, we can share all of this information with the public. Obviously, with a collection this big, it's a slow-going task, but we have several collections that are fully data-based.
Here's one of our imaging systems that we use to digitize our collection. These images we take are both for research purposes as well as just for collections digitization. And they're important to give access to our collection digitally to people and researchers around the world. This imaging system, a Giga macro system, is basically moving from specimen to specimen. We can set up several specimens at a time, and it will photograph all of them, and then we can assemble those images for making a high-quality photograph that we can use for public outreach, for research, as well as just to show what we have in our collection. The images that we have just taken on this imaging system, and some of the ones that my colleagues have taken with different equipment, can be used for research purposes, as well as the public can use them on the Smithsonian Open Access platform or on our entomology collections database.
In 2015, we participated in the museum's first mass digitization pilot project, which was funded by Smithsonian's Digitization Program Office. Our goal was to create a workflow that increased the rate of digitization for pinned insect specimens. We chose the bombus or bumblebee collection due to their charismatic nature and their importance as pollinators. They also lended themselves well to one-shot imaging. In 2019, we revisited this project. we refined our process, completed the Bumbus collection, and also completed the Xylocopa or carpenter bee collection. In total, over 75,000 bees were digitized in just 72 days. Our workflow contained 3 parts: specimen handling, imaging, and data processing. Specimen handling worked like an assembly line. The labels were removed from the pen and placed on the stage. A unique 2D barcode was also added. Then the bumblebee was positioned. The stages were created so that the labels were raised and on the same focal plane as that of the bee. Once imaged, the labels were returned to the pen with the 2D barcode placed upside down so that it could easily be read when removed from the collection. Software was developed to read the 2D barcode in each image, renaming the files accordingly. Standard metadata was also created. These files were then used to create basic catalog records in our collections information system known as EMU. A paid service and Smithsonian Transcription Center volunteers transcribed locality and data label information. This data was then appended back to the catalog records, creating fully digitized specimen label records. This data and their associated images are now available to anyone in the world. No trip to the museum needed.
Digitizing a collection as large as ours is an ongoing process, but some of our groups are fully databased to the specimen level. One of these groups are the Dobson flies, or the Megaloptera, and each one of these large insects has a barcode and is digitized in EMO so that the public can access all of the data.
So in entomology, in addition to insects on pins, insects in fluid, insects on slides, insects for genetic purposes in cryostorage, we have another kind of collection, and that's living collections. In this lab, the Smithsonian Ant Lab, we work on fungus farming ants. These are ants that cultivate fungi for food. They're true agriculturalists, and they've been agriculturalists for over 50 million years. And that's quite a lot longer than humans have been agriculturalists. Right here, we have a nest of a fungus farming ant, a leaf cutter ant, a species of Atta. These are particularly amazing fungus farming ants. They have huge colonies of 5 million or more individuals. And they occur in South America, Central America, and there's a couple of species in the southern United States. These ants go out every day and cut up fresh vegetation like leaves and grasses, and they bring that vegetation back to the nest. They have a huge impact on their local environments. What the ants are doing here in the lab, they're cutting up leaves that we've given them. They're carrying those leaf fragments into the nest. This is a garden chamber here. They have a number of garden chambers. The smaller ants, smaller than the foragers, are processing that fresh vegetation. They're cutting it up into smaller and smaller pieces until they've got like a vegetable paste, which they add to the growing edges of the garden. And then they go off and they find healthy growing mycelium from elsewhere in the garden, and they bring that back and they plant it on the newly added substrate. And then they're constantly patrolling the garden, physically weeding out unwanted fungi and bacteria. In addition to leafcutter ants, we also have nests of a lot of other fungus farming ant species. In some ways, they're not as charismatic. They have very small colonies. The sizes of the workers are very small. The gardens are small. But these are some of the rarest fungus farming ant species that you will ever encounter.
This is the arachnid and myriapod wet collection, and we store our specimens in these drawers and shelves like this, with each vial hopefully identified down to the species level, but sometimes just a family or genus. And these collections are growing all the time. They grow in several different ways, often from donations, like what you see here, which represents 30 years worth of work of students going out and collecting in the Great Smoky Mountains, which we recently acquired. And we also acquire collections when entomologists or arachnologists pass away. And another way that we get collections is by going out and collecting ourselves. So this material was collected in New Zealand. It's part of a long-term study by myself where I'm go out and collect spiders. And what we do is we go out and we wind up with when we come back from New Zealand, our specimens look like this. And then back here at the museum, we sort through all of this material, hopefully down to species level, but sometimes just down to genus or family. And then a lot of this material gets sent to the biorepository. The biorepository is a spot where we can preserve material for DNA sequencing and analysis, and then researchers can use that material. Researchers from all over the world can borrow that material and use it for their own analysis. And we send it in vials like this, where the legs will get sent to the biorepository, and the specimen will ultimately be deposited in our collections as vouchers.
This is another part of the storage here in the entomology department. This is some of our older storage. It also is where we house our true bug collection, as well as our Lepidoptera collection, which includes both moths and butterflies, as well as skippers. This part of the collection is not compactorized, meaning it takes up a lot more physical space, and butterflies and moths in particular take up the most amount of space in the collection because we spread the wings so that we can see both the scale patterns as well as the venation.